Okay, hi everyone. So today is our last lecture of new material for the semester. I'm looking at my calendar here. So today we will wrap up our discussion of chapter 17, that is the role of our immune system in cancer, that we began talking about last lecture. Um, this Wednesday, we will have a review for our third exam. And then our third exam itself, just as a reminder, is next Monday, the 10th, at 12 noon, right? So that's the time that's given to us based upon the UH final exam schedule. Keep in mind that even though it's scheduled at the time of our final exam, it's not a big cumulative final exam, right? It's just the third midterm exam, and it'll be the same format as the previous two. So the third exam will cover chapters 12 through 17. And so it's the stuff, starting from week 12, which was immediately following Good Friday. Okay, so go back and if you don't have the class schedule handy, of course, you can go to La Lima, go in the syllabus tool and get the updated schedule. Okay. I shouldn't say updated because I have not changed this since the beginning, but you might want to take a look to help refresh your memory and remind yourself about the exam time that's coming up on Monday. Also, don't forget, you do have one last quiz. And so after today's lecture, I will make quiz 13 available. And so the quiz 13 is all on this cancer and immune system stuff. And that will be due Thursday night of this week like normal. All right, and that'll be your last quiz. I'll remind you again when we review on Wednesday. Okay, good. Okay, so let's wrap up our discussion about cancer and how important our immune system is in fighting cancer, and also how devious cancer is when you're talking about tumor cells that are constantly mutating, and you get natural selection for cells that have different types of mutations that in many ways can fool or evade a detection from our immune system. So this was the last slide from last time that we did not get to. So just to refresh our memory, make sure we're all on the same page, we were talking about different types of mutations that cancer cells can just randomly get, because the more cancer cells develop, the less stable their genomes become, and more mutations start building up. And the mutations are random. That means a lot of the mutations actually aren't good for the tumor cell. And you get a lot of tumor cells that'll just stop and die because of bad mutations that don't even let them continue as living cells. But every so often, if you're dealing with millions of cells mutating in a tumor, of course, you're going to get a handful that end up with mutations that actually give the cancer cell some kind of a benefit, right? And so either it's a benefit that helps the cancer cell resist something like apoptosis, so the cancer cell doesn't properly die and eliminate itself. Maybe it's a mutation that makes the cancer cell proliferate even more rapidly. Or what we've been talking about is certain mutations that aid the cancer cell by giving it new ways of kind of modulating or counteracting our immune systems. Last time we looked at examples. We said that you might have, for example, mutations that result in cancer cells no longer expressing normal levels of HLA proteins on their surface. So they no longer make normal levels of things like MHC1 platforms, and therefore they stop presenting things from inside of them. Things like mutant peptides, right? Peptides that have mutated amino acid positions in them that normally your immune system would be really good at detecting, right? But we said it's kind of scary to think that, yeah, if a cancer cell stops actually displaying things outside of it, then it can be like invisible to the immune system, right? We also said that mutations might result in the expression of certain types of proteins that end up interfering with the receptors of other immune cells coming down and trying to detect and bind to the cancer cell. And then we did not get to this example here, so we'll start off with this one today. Certain mutations might result in cancer cells expressing things that end up recruiting things like Tregs. So in this cartoon here, right, we have our tumor cell in the middle here, right, this pink thing. And it has a whole bunch of mutations in it. And maybe one of these mutations causes it to express high levels of TGF beta. Right? TGF beta, the purple dot here, is a cytokine that stimulates the recruitment of Treg cells. So this orange CD4 cell is one of those subtypes that we call a regulatory T cell. Remember that the job of the regulatory T cell is it will bind to something using its T cell receptor, and it will send out cytokine messages that actually inhibit or repress the activity of other T cells that come that might otherwise attack this cell. So here you can see that the TGF beta has attracted this orange Treg cell to it. And this Treg cell, it's able to detect that there are strange mutant peptides being expressed on the MHC platforms here. So these other cells, like for example, the cytotoxic T cell comes, right? Its T cell receptors also see that. So normally this cytotoxic T cell might kill this tumor cell, right? Or at least tell it to undergo apoptosis, right? But notice, right, the Treg is sending out things like its own production of TGF beta and production of IL-10. And those signals discourage cytotoxic lymphocytes as well as helper T cells, the Th1 helper T cells, from coming and being able to detect and eliminate this tumor cell. So I think that's a very kind of devious way that the tumor cell is actually using our immune system against us. Right? It's using the normal ability of Treg cells to be able to keep the immune system kind of quiet, because remember, normally Treg cells, they keep the noise level of our immune system down, so you don't have a lot of things activating improperly. Right? But in this case, the tumor cell should be something that you want your immune cells to detect and destroy, but because it learned how to recruit Treg cells, right? The Treg cells then are actually protecting the tumor cell from being destroyed. Okay, good. So let's move on. I want you to consider this. In our discussion of cancer cells kind of fooling the immune system or evading it, at no point in this discussion have we somehow suggested that the immune system itself, there's anything like wrong with it, right? We are not talking about an immune system that is immunocompromised. For example, when we talked about immunodeficiency diseases, certain inherited diseases where maybe you're lacking a particular component of your immune system, so your immune system doesn't work at 100% what it should. Or we looked at things like situations where immunodeficiency came about through exposure to different pathogens, right? Different pathogens like HIV that end up causing the deficiency, for example, by destroying T cells. In the case of cancer, it can be very common that the cancer patient's immune system is actually perfectly fine, right? There's nothing wrong with it. It's just that the cancer cells have come up with ways of avoiding it. Right? So what I want you to consider is that, yes, there are lots of different types of drugs, chemotherapies, things like that you can take for cancer, right? Many of which are compounds or molecules that don't normally belong in our body. And that's why, you take them and they interfere with certain cellular processes that, can prevent cells from dividing, for example. But many of those are poisonous, right? Because many of those don't really discriminate between what's a cancer cell that's dividing and a normal healthy cell that's dividing. That's why lots of chemotherapeutic agents have awful side effects. Right? Lots of those compounds are compounds that interfere with things like the process of mitosis by doing things like interfering with the spindle fiber formation in a cell that's dividing, right? You have the spindle fibers that help pull the replicated chromosomes apart into two daughter cells, right? Certain types of chemotherapeutic compounds interfere with that. But importantly, they don't discriminate between what's a cancer cell that's dividing and what's a normal body cell that's dividing. So as a result, the cells in your body that get hit particularly hard by some of those chemotherapeutic compounds are the ones that are dividing. Remember that most of the cells in our body are fully differentiated, and so they actually don't really divide, right? So those cells maybe don't get hit that hard by certain types of chemotherapies. But there are tissues in your body, of course, that you have things like stem cells that are dividing a lot. Right? So for example, the lining of our gastrointestinal tract has lots of epithelial cells that are constantly being replenished. Right? So, or for example, the hair cells in your hair follicles, right? They're dividing a lot, producing things like hair. Right? So those are the types of cells then that can get hit very hard by these kind of non-discriminatory systemic types of chemotherapeutic drugs. And that's why many of the awful side effects of these of undergoing chemo, and I think you already know this, would be things like losing hair or not being able to eat food normally, right? Because those types of tissues are getting messed up. Right? So those really can be thought of as being poisons, right? And you're really playing sort of this game of chicken where you are poisoning the patient and the patient's cells in their body are dying. But hopefully, right, this is the whole game of chicken here, right? You can actually wait it out and hopefully enough of the cancer cells die, right, before you actually kill the person from the poison. And then you quickly taper off the poison, right? And hopefully then the person's normal healthy cells can then recover. So that might sound very barbaric, and it is barbaric in a way, right? I'm sure like a few decades from now, like your kids and certainly your grandkids, right, when they think about how we used to do chemo for a cancer back in the early 2000s or whatever, right? They'll just think like, wow, what a bunch of barbarians. Like that's modern medicine, or the whole idea of you're playing chicken by poisoning somebody and hopefully you kill their tumor before you kill the rest of them, right? That's why as immunology students, and you've taken a whole semester of immunology now, right, the exciting new frontiers in cancer therapy have to do with simply leveraging the patient's existing immune system, right? Because there's oftentimes nothing wrong with the immune system, right? But getting their immune system to pay attention to the cancer cells. In other words, therapeutically boosting a patient's own immune system to turn on and recognize the cancer cells, right? That has so many more benefits than dealing with these types of chemotherapeutic drugs. Because A, it's what Mother Nature actually designed us to do, right? Mother Nature designed us to have a super powerful immune system that's constantly patrolling and looking for things like cells that are going bad and becoming cancer. So the fact that some of the cells in a patient found a way to get around that, right, doesn't mean that the immune system is somehow any weaker. So if you can activate the immune system and get it to pay attention, that is super strong. And importantly, right, it's natural in the sense that you're not dealing with any kind of weird compounds that are coming in and poisoning, the cells and tissues of your body. So that's why I say, how can we therapeutically boost the immune system to better target and destroy cancer cells? Remember, patient's immune system may be perfectly fine, but the tumor cells have stumbled upon combinations of different mutations that allow them to evade proper immune attack, right? So that's why they very much are like a wolf in sheep's clothing, right? Very hard to spot. So if there's some way to kind of flag or identify these as being the things that your immune system should pay attention to, maybe we can go ahead and actually get the immune system woken up. So hopefully you already know, right, or have a sense of what I'm talking about here. We are talking about antibodies, right? In particular, monoclonal antibodies. So what is monoclonal versus polyclonal? Remember, we learned that when you have an antigen from something like a foreign pathogen presented to your body's tissues for the first time, you will not just have one B cell clone in your body that happens to make an IG that recognizes it, right? You'll have lots, or I should say, relatively speaking, right? A handful of different B cell clones that all have different IG that might be chosen because they all recognize the foreign antigen, maybe a slightly different epitope, something like that, right? So if you were to, let's say, have a situation where you had an animal like a mouse, right, and you were to inject something like, I don't know, small antigens from that represent like antigens that you find from a cancer cell into that mouse's immune system would develop not just one particular antibody, right, but a handful of them. And if you were to harvest those, right, you would end up with a situation where you have a mix of different antibodies that all target the same thing, right? That would be a situation where it's polyclonal. Different individual B cell clones in the mouse each contributed the know-how to make a particular IG. On the one hand, you might say, well, that's good, right? Because that's how a normal natural immune system works. So you have different antibodies that all target the same thing. Yes, that's true. But think about this. If you're going to use those antibodies therapeutically, each antibody represents a different sort of therapeutic agent, right? You don't really know. Some of those antibodies might work better than others. Some of those antibodies, right, might not be super specific to the cancer antigen and might actually also bind to other things in the body, right? And that wouldn't be good. So you're much better off taking a group of antibodies that something like a mouse generates, right, and actually figure out which one of those antibodies really is the best one, right? And then you can do experiments with that antibody to make sure, does that antibody bind to other things that we don't want it to bind to, or is it really specific to the cancer antigen? And if you are happy that that one antibody is the one that you really want to use, forget about the other ones because that just makes things messy, right? Then you can just specify that you're going to produce a lot of that single clone of antibody. And that's what a monoclonal antibody is, right? So the antibodies that we use therapeutically in people that come originally from things like mice that they were raised in, right, that they did their experiments in, right, they are monoclonal, because then you can focus on doing a lot of research to make sure that that one specific antibody does what you expect it to do in people, and it's well tolerated by people, right, and it doesn't have weird side effects. So if we go ahead then and imagine that we identify some sort of antigenic signals on this wolf in sheep's clothing, then we can specifically label this particular wolf here, right? And then the immune system might now pay attention to it because it's coded in antibodies. So monoclonal antibodies can work in different ways. I think the most straight ahead, obvious way that you guys are probably thinking about right now is by opsonizing or coating the surface of a tumor cell with antibodies. Right? That is very much like the way our adaptive immune system should work. Right? It just turns out that the patient himself or herself never gained the know-how to make these antibodies. Right? So that's why we're giving them these antibodies. Right? So this is passive immunity, right? In a way, right? We're passively immunizing them to recognize the tumor cell by putting these antibodies in their body, right? And so here we have a tumor cell, and let's say these light purple little proteins or whatever on the outside of the tumor cell are some sort of antigen that you find more on these tumor cells than you do on normal healthy tissues in the body. Or, right, if you're lucky, this antigen is something that's just specific. Only tumor cells make this particular antigen, right? That would be the best case scenario. So you give this person an IV infusion of high quality a therapeutic monoclonal antibody that's been designed or chosen because it recognizes that particular tumor cell antigen, right? So the tumor cell now gets covered up by these antibodies, and the stems of these IgGs are what the FC epsilon or FC gamma, FC gamma receptor on NK cells, natural killer cells, will recognize, right? So FC gamma receptor binds to the stem of the antibody, and now this natural killer cell that up until this point was just content floating on by, right, or even bouncing off this tumor cell and not seeing anything on the tumor cell that it cares about, now the natural killer cell will pay attention, right? And so the natural killer cell will then release its cytotoxic messages and tell the tumor cell to undergo apoptosis. Keep in mind that if you're dealing with antibodies, right, this would not be a way of recruiting T cells, like alpha beta T cells would not be recruited in this manner to this tumor cell. Why? Because remember, the alpha beta tumor cells of your immune system, they need to be presented things as small peptides on MHC, right? They're not going to care that there are stems of antibodies sticking out from the surface of this tumor cell. Right? But there are other ways that we can recruit T cells as well, or increase the activity of T cells. So we'll take a look at that as well. Right. One of the ways that antibodies can also activate things like T cells, right, is by increasing the strength of T cell responses. So you'll remember that a naive T cell, let's look at this first panel of the cartoon here. So the blue is a naive T cell. It has its T cell receptor, right, that recognizes a combination of some kind of a red dot here, this peptide on MHC class 1, right? So this is a cytotoxic T cell. And remember, it has CD28 that needs to see the B7 signal from a professional antigen presenting cell. And so this might be, for example, a dendritic cell that is picking up different bits and pieces of strange mutant peptides from some tumor cells, right? And it's presenting this to the T cell up here. And this T cell happens to have the right T cell receptor shape to recognize it. So not only does the T cell receptor signal stimulate the T cell to activate, but of course the B7 CD28 is that extra message of high confidence, right, that this T cell should activate against this message. One thing that we didn't discuss is, like other examples of cells in the body, right, we said that a lot of cell signaling is always this kind of tug of war between stimulatory signals and inhibitory signals, right? That's certainly the case with natural killer cells activating. And, one thing that we didn't really explore is, to a certain extent, that's also true with the T cells. So T cells not only have the CD28 signal waiting to see the B7 salesperson badge being presented by the antigen presenting cell, like the dendritic cell here, but its surface also has this other receptor known as CTLA4. CTLA4 also binds to B7, but in this case, CTLA4, right, does not stimulate the activation of the T cell. So in other words, this CTLA4 is another way of keeping the noise level down, right? Because if the CTLA4 preferentially binds the B7 signal, then this naive T cell never gets the B7 CD28 signal, right? So that's a normal way that our immune system sort of limits the number of T cells that actually wake up and get active. So in the case of cancer, right, you don't want those limitations, right? You want every T cell that recognizes the tumor peptide to be able to fully activate, right? This is not a time for playing games with keeping the noise level down. Right? So you can use an antibody. Look at this here. This yellow antibody is an anti-CTLA4 antibody, a monoclonal antibody that was designed to bind to CTLA4. And by binding to CTLA4, right, it prevents CTLA4 from being able to interfere with the B7 CD28 signaling. So you get more and more T cells that recognize the red dot here and then are encouraged to activate. Right? So this is an example where the antibody is not working to somehow recruit additional immune cells by its stem, It is working just to go ahead and interfere with things like the CTLA4 receptor. All right, so existing T cell sensitivity is greatly enhanced in this way by the addition of this antibody. Another way that antibodies work that is not that obvious, but hopefully when you see this, you'll think this is really clever, right? Is you can conjugate things to the antibodies. Remember, antibodies are really just small proteins, and we can link all kinds of things to proteins. So here in this case, there is a cytotoxic drug, this auristatin, this gray ball over here, right, which is toxic. It's toxic because if this gets into cells, it interferes with the microtubule formation, and therefore the cell cannot properly make a spindle fibers, and it won't be able to undergo mitosis properly. Right. So normally auristatin, if it's given systemically, meaning just the naked drug, it is really toxic, right? Because all the cells in your body that get exposed to the auristatin, right, would have some sort of nasty impact on their ability to divide, right? But what if you link or conjugate the auristatin to a specific antibody that you've chosen because that antibody is specific for some sort of antigenic signal that only the tumor cells have, right? Now what you see is that in this case here, this is a lymphoma cell, right? So some sort of a B cell cancer, and on its surface, it has CD30. So these red triangles are CD30, something that these lymphocytes would have, right? That other types of cells in your body wouldn't necessarily have. So now you give this person an infusion, an IV infusion of the specially made conjugated antibody that has the auristatin in it. And the antibody floats through the blood. Fortunately, right, you're giving very low levels of this compound, so there's not a lot of toxicity. But what? When the antibody encounters one of these lymphoma cells that has the red triangles, the CD30 on its surface, right, the antibodies will bind to it. So the whole outer part of this lymphoma cell will get covered by this IgG. And as a result, when they get taken in, right, the dose of auristatin that this particular cancer cell gets because of all these antibodies bringing it to it, right, is way, way higher dose than the normal healthy cells of your body would be exposed to the auristatin, right? So that's a way of actually using antibodies to deliver high effective localized doses of chemotoxic agents and sparing other tissue. Right? Another way you can do it is you can conjugate things like radioactive isotopes. Right? So in this case, the little red asterisk looking thing is a radionucleotide that's again, right, conjugated or attached to this antibody. And as a result, only the cells in the body, like the tumor cells that express the CD30 here that the antibody recognizes, only they will get the lethal dose of radiation, right? Because the other cells in your body, right, if these antibodies don't bind to them, the effective dose of the radiation floating by them is too low to cause damage, right? But if you have a cell like a lymphoma cell that gets covered with these antibodies, and those antibodies are all tethered to the radioisotope, then that particular cell gets a much higher lethal dose of radiation, right, that destroys it. So this is what I mean. In some ways, this seems remarkably simple in concept, right? But I think in other ways, you think this is a really wonderful, clever way of using antibodies. That is a way that, you know, is not the normal way you think of antibodies functioning. So here's an example, a table of a number of examples of different types of monoclonal antibodies that are now being used in treating cancer. And you can see this ranges from breast cancers to colon cancers, squamous cell carcinomas, melanomas, colon cancer, B-cell lymphomas, leukemias, etc. These are the particular monoclonal antibodies. And you probably already have seen that therapeutic antibodies have very funny names to them, like trastuzumab or cetuximab or ipilimumab or rituximab, right? And you always, and maybe you've thought like, who makes up these names, right? Well, it turns out that there is a sort of methodology to this kind of madness. Right? Hopefully you now realize when you see a commercial for some sort of a drug on TV, here in the US, we get tons of drug commercials on TV, right? And you see that it ends in MAB, now you know what MAB stands for, right? This is a monoclonal antibody drug. And so anything that ends in MAB, you now know is a therapeutic antibody, monoclonal. Other parts of the name, like the ZOO or the XI, right? These also refer to different ways that these antibodies were first isolated, whether they were isolated in mice, for example, or if they're based upon a mouse antibody that's then been sort of recombined with a human antibody, right? That's all part of the name as well. Okay. So these naked antibodies are straight up working like regular IgGs, right? And so you can see they work to block or interfere with different types of signaling that are that otherwise would encourage cancer cells to keep dividing and growing. And again, these all work by binding specific epitopes in a way to prevent something else from binding there. For example, being able to bind something on a receptor that prevents the receptor from normally receiving the message that would stimulate the cancer cell to grow. The two on the bottom here are the real-world examples of two different monoclonal antibodies that fit what we just talked about on the previous slide, right? The brentuximab is the one that's conjugated to the orostatin, the microtubule cytotoxin. Right? And then here you have this radioisotope labeled monoclonal antibody that's used to treat lymphoma. Okay. So here's a question then. A big part of our discussion this semester about adaptive immune response had to do with understanding and appreciating vaccines, right? And understanding the principle of vaccines. Remember, you can boil it all down to the principle of vaccination is to be able to give your immune system a preview, right? Introduce your immune system to different antigens that are found or represent stuff that would be on the surface of a particularly bad, nasty virus or bacteria, something like that. And so by giving your immune system a preview of that, you have the opportunity for your adaptive immune system to develop B cells, T cells, immunoglobulins, antibodies that have memory, right? So that when the real virus comes, you already have the ability to have a strong secondary immune response. So can you vaccinate against cancer? That's an interesting question, because after all, cancer cells are not foreign pathogenic cells. They are cells from within the body's normal tissues that have just over time gone bad, right? Or gone rogue, as I like to think of it, right? Through a series of random mutations. So how do you vaccinate against that? Well, there are antigens presented by them, right? So can you actually utilize that? Here's another way of thinking about it. When we talk about vaccinations, we usually think about things like viruses, right? So are there certain viruses that are related to cancer? Yes. I think we mentioned last time, right, that about 1/5 of all cancer types have some evidence of being linked to some kind of pathogenic infection. We looked at several examples last time. I think one of the most compelling and poignant one is the type 1 diabetes, right? Because we now believe that type 1 diabetes in little kids can be linked to infection by Coxsackie virus, this very common type of virus that kids transmit to each other in nursery school, preschool, right? And in some kids, unfortunately, that Coxsackie viral infection doesn't just end with a few days of maybe some fever or cold-like symptoms, right? In some kids, that ends up triggering autoimmune reactions, right? Where the immune system then begins to see things that look like the antigen being made by cells of the body, like the pancreas, right? So can we vaccinate against things like cancer? Here's an example. Another type of cancer that there is an undeniable link between cancer and viral infection, right, has to do with human papillomavirus, or HPV. So definitely HPV is linked to the formation of cancer. HPV ranks as one of the most common causes of sexually transmitted diseases. It's been estimated that almost half of all sexually active individuals have at some point been exposed to HPV. Right? This doesn't mean, just because you're exposed to the virus, that you necessarily ever had any symptoms of viral infection. Right? The majority of HPV infections usually entirely clear from the system in 1 to 2 years. However, remember, a virus normally infects a cell and hijacks the cell's machinery so that the cell is basically forced to make more copies of the virus particle. And when those virus particles are released, of course, it infects other cells, and the original cell dies in the process because it basically gets worn out just making all these viral particles. Right? In some cases, though, the viral DNA doesn't succeed in properly integrating into the human genome in a spot where it can carry out its hijacking functions. But because it does randomly integrate in the genome, right, it does mess things up. So for example, maybe the viral DNA integrates in places that disrupts the sequence of a normal gene. Maybe it's a normal gene that codes for one of those proto-oncogenes that drives the cell cycle forward, or maybe it's one of those genes that acts as a tumor suppressor that normally is able to detect and stop a cell from dividing out of control. Right? Either way, that type or that instance of viral integration of the genome may actually lead to that cell transforming and becoming cancer. And so HPV infection is most prominently linked to things like cervical cancer, right, other genital cancers, and in some cases, throat cancers in men, right, or in males. So here you have a picture of the HPV particle, I'm sorry, right? And you can see here, this is illustrating that you have virus particles infecting the cells. Many of the cells are hijacked, and so they just produce lots of virus particles. Right? In some ways, these are not the cells that you worry about, because if the cell is hijacked, it's just eventually going to die and not stick around, right? But the ones that you worry about are the ones where the integrated viral DNA, right, does not succeed in fully hijacking the cell, but it just incorporates in the cell and sticks around, right? And any types of mutations that causes could potentially lead to the cell transforming. What does this mean? If HPV is the leading viral cause of cervical cancer, and HPV is a virus, then what if you can vaccinate against HPV? You potentially could eliminate the dominant cause of cervical cancer. Right? And so that's what this vaccine is. So Gardasil, which is the human HPV vaccine, right, has been shown to be tremendously effective, not only in preventing HPV infection, but also, because of preventing HPV infection, being tremendously effective in preventing precancerous lesions from forming on the cervix. In fact, in studies, so Gardasil is based upon, this table here shows that there are these different sort of families of proteins on the surface and inside of the HPV virus particle, right? Some of these things are different transcription factors inside of it. Other things are part of the outer capsule. So not surprisingly then, the proteins that are part of the outer capsule, those are the ones that are really good candidates to use as antigens for developing antibodies against, right? Because they're on the outside of the virus. So this particular protein, this L1, is this major capsid protein. And what researchers have done is they've looked at 9 different strains of human papillomavirus and looked at the L1 protein that's on those. And these are the strains that are the most highest linked to cervical cancer. And so the HPV vaccine then is a multivalent vaccine that presents these different antigens to the immune system. So they've now shown that Gardasil 9, that's what it's called, that's the market name for it, is 100% effective, right, in preventing these lesions of the cervix, right? So in other words, it's that effective in protecting against cervical cancer, just because it's able to prevent infection by the virus that causes cervical cancer. It's controversial, though, because this is a vaccine. So just like any other vaccine, right, it's the most useful only if it's given before the individual has been exposed to HPV at all, right? That makes sense. And because we're talking about kids, the recommended age for Gardasil or the anti-HPV vaccination is now 9 years old, right? Because they want to make sure to get to the kids before they become teenagers and before they are exposed to HPV. So it's controversial because a lot of parents don't like the idea that they're vaccinating their 9-year-old against something that's protecting them against things that they might get when they're sexually active, right? Because they don't like thinking about their 9-year-old that way, right? But the truth is, the general public should be made aware at how effective this is because you're protecting your child against the development of certain types of cancers in the future by getting this vaccination. Here's another example of a very, very clever way of introducing or letting your immune system see the cancer that it was previously ignoring. This is a treatment strategy that's also in a way considered a vaccine, but you'll see it's kind of different than traditional vaccines. And this is designed to fight against prostate cancer. So this slide here shows various prostate tumor cells, and the pink or red stain is staining for a specific protein that prostate tumor cells express on their outside, right, that other cells in the body do not. And this is called prostatic acid phosphatase, or PAP. So because the prostate tumor cells specifically express PAP, you might think, hey, that's a good candidate as an antigen, right, if you want to specifically target these cells. So what researchers do is they've produced an artificial ligand here that is the PAP protein or peptide from the PAP, so the red diamond here, and they've fused it to another protein. This is what we call granulocyte macrophage colony stimulating factor, or CSF. In other words, this is a normal cytokine message that certain immune cells are designed to very greedily take up. Okay. You can see here this ligand, so they call it this PA2024, that has 2 parts to it. The blue part, which is the colony stimulating factor, right, gets very, very easily taken up by things like monocytes. So monocytes have this green and yellow receptor on their surface, right, and it'll bind to the blue part, right, but it'll go ahead and it'll activate things like these monocytes to activate and become macrophages. But the real immune cells that this therapy relies on are our dendritic cells. So here's a dendritic cell. It also binds to the colony stimulating factor on its surface, right? But what do dendritic cells like to do? They love to take all kinds of things in, right, and process it so they can display things on the outside. So these dendritic cells then, right, taking this in, will go ahead and present that red peptide. So how does this work? The way this works is that a patient, so a man with advanced prostate cancer, right, maybe this is a cancer that is no longer surgically operative because they've maybe already tried. They've already removed the prostate, but they were leftover cancer cells, and those cancer cells have now divided and the tumor is recurring. Right? So the man will go in and they'll actually take from his blood, right? They'll take blood out of him and they will purify the dendritic cells, his own dendritic cells from his own blood. They'll essentially put it into like a test tube, right? It's a little bit more complicated than that, but you can think about it as a test tube, and they will force feed those dendritic cells this PA2024 ligand, right? And those dendritic cells will take a lot of this up because the dendritic cells, again, they love the blue dot part of this, right? Because they're designed to take in this colony stimulating factor, right? So boy, these dendritic cells in the test tube then, right, as they're processing all these proteins, they are displaying then tons and tons of the red dot, the PAP on it, right? That's what this third and fourth cartoon is showing here. So all of this stuff so far, you're like, yeah, we've learned about this, right? The dendritic cell takes stuff in, processes, and then presents on MHC. So then what they do is the man comes back into the clinic and they put these dendritic cells that have been trained now, right, to display tons of the PAP peptide, they put them back into his body, right? And then they go into his lymph nodes and they go back to work, and all these T cells are coming to see what's going on. And essentially now these trained dendritic cells are saying, all right, forget about all the stuff that we normally see, that we normally show you. This red dot, right, that we are loading up on all of our MHCs, this is what we need to pay attention to. And of course, then you select for the T cells that previously, right, were not being activated to recognize this. Now they will activate and they will go to the man's body, through the man's body, and back to where these prostate tumor cells are. And now these T cells will start attacking the tumor cells, where previously, right, the immune system was leaving them alone. So I think hopefully you find that really exciting. But the drawback of this, right, is the expense, right? Because we're not talking about just purifying some antibodies and giving them as an IV. This is a whole process of taking blood out, purifying dendritic cells in the lab, training these dendritic cells to preferentially express a peptide that you want them to, and then putting these back into the body, right? So currently, this type of treatment, it's done in 3 visits to the clinic over the course of a few months, and the cost is about $30,000 per visit, right? So the whole Provenge course of treatment is about $100,000. Right? And at this point, I don't think the insurance companies are all that keen on paying for it because it's still considered sort of an experimental type of treatment. Right? But, you know, in the future, maybe 10 years from now, 20 years from now, this type of immune cell training, right, might become cheap enough that this becomes a very routine type of treatment strategy. And the benefit, of course, is that it's hugely powerful. Again, why? Because you are leveraging the patient's own immune system, right, getting it to wake up and pay attention to the cancer when it just previously wasn't, right? And because these dendritic cells were taken from the same body that they're put back in, there is no worry about things like HLA matching, right? That might lead to things like tissue graft transplantation rejection. Right? Very powerful, very exciting, and it's bound to get cheaper and cheaper as time goes on. Okay. All right, so to wrap up then, you know, I think this semester we've talked a lot about the immune system and the functions of the immune system in many different sort of lights, right? Different ways that the immune system works to protect us. I think when we started the semester, you probably just thought of the immune system as protecting you against things like pathogens, things invading you from outside. Okay. Remember, I think our very first lecture, I was telling you about how we take for granted our immune system and how we don't think about our immune system unless we get sick, and then we kind of blame our immune system for being weak, right? How come I get sick when other people don't get sick? When in reality, I told you from day one of this class, right? If your immune system wasn't strong and it didn't work, right, you would not have made it this far in your life. Right? But importantly now, by the time we've gotten to the end of the semester, hopefully you've expanded your appreciation for what the immune system does. Not only does our immune system protect us against things that are outside, right, external threats, but now you realize, just as important, if not more, every single day, your immune system is protecting you from your own cells, the trillions upon trillions of cells in your body, right, that every day have the potential to start making mistakes and going bad and leading to things like cancer. All right, so that's it for the semester. Thank you all for a really wonderful semester. You know, I'm always bummed that we didn't have in-person class, but you know, I mean, I think the online teaching did have some benefits. For example, the YouTube videos that I know, being for many of you making use of the YouTube videos to really be able to go over stuff. But in many ways, to me, that doesn't replace the fact that I didn't get to see all of you in the classroom. Right. So for some of you, I did get to know, right? And so that's wonderful. I continue to encourage you to, you know, let me know how things are going with your other classes. For those of you who I didn't get a chance to meet, hopefully, you know, as our year goes by and we head into the fall and more people are on campus, I certainly encourage you to stop by, say hello. If you ever see me walking around, say hi. Let me know that you're one of my immunology students. I'd be really happy to know that. All right. But thank you again. So I'll see you again on Wednesday when we'll review for our last exam. And in the meantime, I'll go ahead and activate the quiz on the cancer stuff so that you can remember to take that in time. All right. And it's due this Thursday night as usual. All right. All right. So I'm going to stop there.